Hello, everyone, and welcome. Today's live webinar, Choroid Plexus Epithelial Cell 2D and Modified 3D Cell Culture Model, is presented by Dr. Elizabeth Delery, Postdoctoral Fellow, Department of Physiology, Louisiana State University Health Sciences Center. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, and it's brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com/cell culture heroes. Now let's get started. I'd like to inform everyone that today's webinar is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing the presentation, just click on the Support tab found at the top right of your presentation window, or you can let us know you're having a problem by using the Ask a Question box at the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Elizabeth Delery, Postdoctoral Fellow, Department of Physiology, Louisiana State University Health Sciences Center. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Delery, you may now begin your presentation.
Thank you, Christy. Hello, everyone, and thank you for joining me today. My name is Dr. Elizabeth Delery, and today I will be presenting my Choroid Plexus Epithelial Cell 2D and Modified 3D Cell Culture Model. So today, this is just my overview on what I'm going to be going through, just a little bit about me, a little bit about HIV, and then leading into why I wanted to look at the choroid plexus and just a little bit about that, just for background for those of you that aren't aware of it, as well as moving into how I designed a 2D cell culture, and then how I created my modified 3D model. So about me, as I said before, my name is Elizabeth, and I'm a postdoc in the Physiology Department at Louisiana State University Health Sciences Center. Currently, I study the neurobiology of alcohol, opioids, and pain. I attended Randolph College for my undergraduate studies, where I majored in biology and psychology with a pre-med concentration. It was while I was here that I was first introduced to research, as you can see from the little blue square in the bottom left. I worked in the biology department, where I studied soil characteristics on Listeria inocua survival and growth. In the psychology department, I actually looked at various exercises and social activities on cortisol levels and expression. So I really decided that undergrad really introduced me to research and wanting to figure things out. So my professors encouraged me to go on and pursue my PhD. I was accepted to Tulane University School of Medicine Biomedical Sciences PhD program, and I completed my PhD at Tulane National Primate Research Center. My dissertation was actually on the choroid plexus and HIV pathogenesis, and I just graduated this past spring. In my free time, my husband and I like to go on adventures, and we have two very rambunctious dogs, Wishbone, which is on the left, and then Thor, who's the little goofball on the right. Also, in my free time, I run an Instagram page where I like to talk about science. I like science communication because I like to help fight the internet pseudoscience that's out there, as well as showing that science is fun and get people excited to learn about it. I also want to break the scientist stereotype that we're always in the lab and that we're really boring and that we don't have lives outside of science. And then I also like to promote inclusion in STEM, whether it's race, sex, orientation, disability, etc. And here are some examples of some goofy posts I've posted. The one on the left, I was talking about eyeballs and how we see with some super goofy eyeball glasses. And the other one was a maternity photo shoot I took with my dissertation because it took me about 9 months to write. So let's start talking about my research. So just in general, a little background on the HIV epidemic. In 2017, there were 36.9 million people living with HIV in the world. They had around 1.8 million new diagnoses and just shy of 1 million deaths from HIV-related disorders in 2017. So it's still a very pertinent global health issue. In the United States, DC, oops, moved too far forward. In the United States, DC has the highest percentage of new cases of HIV in the United States, and then Louisiana, which is the state that I currently live in, is actually in 4th place with 22.1 new diagnoses per 100,000 people. So it's still very close to home and a very important issue for me living in Louisiana. So some primary obstacles to curing HIV are the presence of long-lived viral reservoirs. Now, these reservoirs can persist despite the use of long-term combination antiretroviral therapy, which is the drugs we use to treat HIV, and that's combination, 2 or more drugs taken at the same time. The brain is one of these long-lived viral reservoirs. Now, since my background at Randolph was biology and psychology degree, really wanted to focus on the brain and figure out what was going on and why issues with the brain being a reservoir. So when HIV infects the brain, it leads to a series of disorders known as HIV-Associated Neurocognitive disorders. There are 3 different levels, and here's the list of diagnostic criteria on how to identify. As you can see, you have asymptomatic, which is more of the mild. It has the prevalence percentages in the center column. There's around 30%, and that's in combination antiretroviral treated HIV-positive individuals. You also have mild, which is a slightly more severe type, and then the most severe is actually HIV-associated dementia. Collectively, these disorders are known as HAND. That's just a little name for them, acronym for them. So HIV is believed to enter the central nervous system when infected monocytes or macrophages are unknowingly trafficked across the central nervous system via a Trojan horse or Trojan herd model. The Trojan horse model argues that a few infected monocytes and macrophages enter the central nervous system, and they establish a latent infection in the brain. While the Trojan herd model argues that there's a surge of infected monocytes and macrophages into the central nervous system late in infection towards the progression to AIDS. Either way, when HIV enters the central nervous system, it can lead to these disorders. So pathologically, HIV, when we're looking at like the actual slides of brain tissue, neuropathology includes lesions, perivascular cuffing, leukocytic infiltrates, and the presence of multinucleated giant cells, which are these giant cells that are a result of macrophages fusing together in chronic inflammation, whether it's a failure of division or whatever. They just make these gigantic cells. And as you can see on the left, I just have an example of an H&E of one that 2 of the vets at the primate center took, and it's kind of towards the top area. You see this big circle with a lot of blue nuclei around it, or purple circle with a blue nuclei around it, and that's your multinucleated giant cell. And for a better example on the right, this is an immunofluorescent image of a multinucleated giant cell, which is pretty much dead center in the picture. And it's a green circle with the blue dappy nuclei around it. The green is CD163, a macrophage marker, because again, these cells are formed from macrophages. So in doing a literature review to learn more about HIV-associated neurocognitive disorders, I began to focus on the choroid plexus, which is the blood-cerebrospinal fluid barrier, CSF barrier, a place where HIV-infected cells can enter the central nervous system, and it's been identified in the literature as being one of the reservoirs. And it's also been identified as being an area of productive infection as well. So the choroid plexus lines the lateral third and fourth ventricles in the brain, which is like a space surrounding the brain. As you can see, kind of like the inner side of the brain. I can't really point, but you've got the arrow on the top right, if you can see, right below arachnoid. You see the choroid plexus in the center kind of the skull. It produces the here's a better picture of it, how it lines the actual ventricles. And it produces the cerebrospinal fluid that protects the brain, and it's composed of these fenestrated capillaries inside a neuron-devoid stroma surrounded by a single layer of epithelial cells. So that's how you really identify it. It kind of looks like this curly, like broccoli almost, but it's got a single layer of epithelial cells on the outside, and that's really how you can find it on a slide. It's super, super easy to spot once you know what you're looking for. So I've got some more pictures so you can get a better idea of what it looks like. This is kind of a 3D representation of it. As you can see, again, I described it like broccoli. Doesn't really look like broccoli, but you've got these protrusions that come off of it. So it's this 3D structure that's kind of floating inside the ventricles. It's attached, but floats out into the cerebrospinal fluid that it produces. And then here is a better example of the single layer epithelial cells, followed by, you have the inside, the fenestrated capillaries on the inside. It's got the endothelial cells surrounding the capillaries. So for something to get into the central nervous system, it has to first cross the endothelial cells of the fenestrated capillaries. It has to encounter any immune cells inside of it, so like macrophages or microglia, and then it will travel out through the choroidal epithelium. It allows for the diffusion of water and sodium through to maintain like water homeostasis in the brain, and it can also allow the movement of larger molecules like glucose into and out of the brain. Like I mentioned before, cells can traffic through the choroid plexus and basically a normal immune surveillance, but you can have increased movement in times of brain damage. It can also prevent drugs or allow drugs to enter the central nervous system because it has multi-drug resistant proteins to either block or allow drugs from entering the central nervous system. So just for a brief history of the background of the choroid plexus, it was first identified by Herophilus way, way, way back then in BC, and he called it a choroid concatenation. Galen later in like 130 to 200 AD first gave it its true name of choroid plexuses. It was discovered in 1543 by Vesalius that choroid plexus produces cerebrospinal fluid. But it really wasn't until the early 1900s that you started to get the detailed structure and morphology. You know, with increased scientific technology and microscopes, we were able to actually really see more about the structure and how it's set up in the brain. If you want to know a little bit more about this and just some background on it, Shane Littlelow has two really great review papers that are actually really interesting reads out there. That just the development of the choroid plexus and the CSF barrier, as well as just the different barriers of the brain and just how we've learned about it over time. So those are both very, very great review articles if you'd like to go back and read more about this topic. Now, I've been boring you about the background research. You really want to know why the choroid plexus and why we wanted to focus on this. The choroid plexus is an adult stem cell niche. It's one of the locations that's been known to create neural progenitor cells. It's the interface between the blood and the cerebrospinal fluid, or CSF. It allows a transmigration of monocytes and leukocytes into the central nervous system, and it's responsible for immune signaling. It can recruit monocytes and leukocytes in infection, hemorrhage, and stroke. So if you have brain damage, it can actually signal out to the peripheral immune system and request additional cells to come help out with any damage in the brain. It's also responsible for the diffusion and transport of drugs, as I previously mentioned. And like I said from some of our earlier work, it's a reservoir for HIV. Since HIV-associated neurocognitive disorders are the result of infected cells entering the central nervous system, whether it's via Trojan herd or Trojan horse model, having a choroid plexus epithelial cell model would allow us to study the barrier properties at one of the last lines of defense between the blood and the cerebrospinal fluid in the central nervous system. So that's kind of what got me interested in the choroid plexus and studying it and figuring out why. So, like I said, we sought to figure out a way to create a choroid plexus epithelial cell culture. Now, when you're designing a primary cell culture, you have to keep a lot of things in mind. You want to know what cells you're looking for. You have a couple different types: epithelial-like, fibroblast-like, lymphoblast-like, and those types all have different needs in culture. You also need to know the type of substrate, whether they can just sit on the plastic petri dishes or if you need gelatin, collagen, or matrigel. You also have to use sterile conditions when you're using your cell culture to prevent bacterial and fungal contamination, which can completely ruin your culture. You also need to know what type of food or media the cells need in order to grow and survive. These can include things like different amino acids, carbohydrates, vitamins and minerals, growth factors to help promote growth, and even hormones. And also you need to have culture conditions, like when they're in an incubator, what concentration of gases do you need, pH, osmotic pressure, and even the temperature the cells need to grow. So there are a lot of different factors to think about. Now, if you're curious about this and you want to learn more, GIBCO has some great background stuff. They have the Cell Culture Basics Handbook and an online interactive lab, which are both free and awesome, and I actually used both when I first started out trying to figure out how to design my own cell culture. So to get practice before you actually get in the lab and do stuff, I do highly recommend these resources. So since we knew we wanted epithelial cells, they're polygonal in shape and they need a substrate to attach to. So we looked at the 3 different types. These are just some goofy pictures I got. We have gelatin. This is a picture of Jell-O because it's the closest thing. Gelatin is just sort of this clear, boring goo. So I figured a picture of Jell-O was a lot more fun. You also have collagen. So here's a picture of collagen matrix. And then here's a picture of the actual matrix to symbolize matrigel, which is the more extreme, really nice substrate with which to grow cell culture. So since our lab already does a lot of cell culture work, we grow both brain microvascular endothelial cells and astrocytes. We kind of had an idea of what media we're going to need to grow brain cells. And after a lot of playing around with different types of media, like DMEM and M199, we settled on M199. We added some additional L-glutamine, which is a, losing my spot. It's an amino acid energy source for the cell, and you need that for them to grow. And M199 also has aryl salts and a phenol red pH indicator, so you can follow the pH in the cells and see if the cultures are going well. So that's why we chose M199. And we also added an anti-anti, which is an antimycotic and antibacterial thing to help prevent the growth of contaminants in our cell culture, which again would completely ruin them. And then we also used fetal bovine serum. We used exosome depleted fetal bovine serum to have a really pure FBS to add to our cultures to help the cells grow. There's a lot of trial and error involved in this culture, and I killed several different cultures before we actually got them fully started. But once we figured out the ideal substrate and growing conditions and the media and additional supplements we needed to grow, and about the first week after plating the cells, after digestion, we started to see the cells attach and colonies begin to form. And as you can see on the left, you've got a colony of about 5 cuboidal-like cells that are forming a little baby colony. And on the right, it's a little bit harder to see, but you have a slightly larger colony. There was a lot of inter-animal differences depending on the seeding density with which you plated the initial cells, how long it took them to attach, like 24 to 48 hours to the collagen substrate, could all affect how quickly these cultures grew. Now, in 2 to 3 weeks, you start to see these colonies expand and spread out. And again, they're polygonal, cuboidal-like cells, which are definitely epithelial-like in nature. So it's kind of what we were expecting to see in our cultures. It took about 4 to 8 weeks, again, depending on animal variation, seeding density, things like that when we first started. But within 4 to 8 weeks, the place began to become confluent. We were worried a little bit about fibroblast contamination, as you will with most cell cultures. So if you saw anything that started to kind of look like fibroblasts growing in the cultures, we treated with something called cis-4-hydroxydiproline, or cis-HP, which has previously been published to be a fibroblast treatment. So we use that if necessary to take care of fibroblast contamination. So once you reach confluence, the next step is to see, hey, this is a primary cell culture. Am I able to subculture this and keep dividing it and make more wells of it? So we were able to successfully subculture. You can kind of see in the top left how there's that like little corner. That's because this is a collagen-coated little disc that we put inside the wells so that we could grow the cells on these discs, and then we could fix them and then stain them to confirm the identity. Because yes, these cells are epithelial-like in nature, we can't conclusively say that they're choroid plexus epithelial cells at this point. So the next thing we had to do, again, validate them and confirm their identity. Now we can do this by finding literature-reported markers for the cells of choice. So we used clotho, which is an anti-aging hormone, transthyretin, which is a binding protein that transports thyroxine and retinol, and the capitalized letters are how it got its name. We also used vimentin and phalloidin. Vimentin is an intermediate filament protein, and phalloidin stains F-actin. So those are both kind of like structural proteins to, really show that we've got this cuboidal and polygonal-like cell. And we also use zonula occludens 1, or ZO1, which identifies tight junction proteins. Now, if you remember from the choroid plexus pictures from way back, choroid plexus is also known for having tight junctions between the single-layer epithelial cell around the outside. So ideally, we would like to see ZO1 in these cultures. We also want to look at focal adhesion kinase, which identifies cell-to-cell adhesions. You also have to do true staining to say, hey, what I've got is correct. You also want to do isotype controls to make sure there's no non-specific binding of your antibodies to anything in your cells. So you always do isotype controls to make sure that the staining you're seeing from all the above antibodies are true. You also want to exclude other cell types when you're confirming your identity, like neurons, astrocytes, microglia, and even macrophages. Now, thankfully for us, there are no neurons in the choroid plexus, so that ruled out that cell type. But you also have astrocytes, so we stained for GFAP, microglia, which we looked for IBA1, as well as macrophages for CD163. So this is how you would go through to confirm, to say that, hey, we've got the cells of the shape that we want, the morphology that we want. We think these are it. Let's confirm by making sure it stains for true markers. The markers are true staining, as in it doesn't bind to non-specific binding in the background, and then you also exclude other cell types. And then at that point, you're kind of like narrowing it down to say, hey, this is the cell type that I want, and I've got the correct type. So here's an example of our clotho staining, and again, clotho is the anti-aging hormone. I've got the legend on the right listing the different colors. Didn't think about this ahead of time, but I do apologize if you are red, green, colorblind, because I put clotho and phalloidin in red and green. So I do apologize about that. But clotho is the red stain that is paranuclear to the blue DAPI. And then phalloidin, again, is the structural protein that kind of just shows the shape of these cells. And as you can see, all of the cells here were positive for both clotho and DAPI, sorry, phalloidin and DAPI. So you do see the cells, and they are positive for clotho and phalloidin, which just kind of give you the shape and also show you that they are positive for a marker of the choroid plexus. And next, we looked at transthyretin and DAPI. It's a little bit more of a fainter stain on the screen, but again, with the isotype controls, this was true staining. As you can see here, the cells were positive for transthyretin, which is in green, and then the DAPI in blue. So you can see the kind of, again, cuboidal cells that are positive for transthyretin. They've got their little DAPI nuclei in the center. Then we looked at the structural proteins again. You've got the vimentin and the phalloidin. Phalloidin, again, is in blue, so kind of the blue squares is the phalloidin staining. Again, the DAPI in the center is the nuclei circles. And the vimentin is red. So you can kind of see the structure and the shape and even the actin and the filaments in these cells, which is super, super cool. Here was my combined image with both the transthyretin, vimentin, phalloidin, and DAPI. It's one of my favorite pictures because you have that nice cuboidal cell in the center of the picture. So you've got all the positives for the stainings that we're looking for, plus it's got the shape. It's really looking like we had identified choroid plexus epithelial cells. So the next thing we wanted to do was look at the ZO1, or the tight junction proteins. And as you can see here, I had to increase the brightness just so you could see it just a little bit better on the screen, since it is tinier with the PowerPoint. You can see the DAPI nuclei in the center, and as you can look around a lot of these cells, you see these bright red kind of dots. If you're able to zoom in, you see these nice lines, and this is indicating the tight junctions that are forming between the cells. That's ZO1 staining. So these cells had formed tight junctions with each other, again indicating choroid plexus epithelial-like features. And then again, we looked at focal adhesion kinase. If you kind of look to the bottom left, you can see a really great example of two, where you see those little like red spikes coming off the cells, and that's actually where they've formed adhesions or are trying to form adhesions with adjacent cells. So as of right now, they are all positive for all of it. So using all this knowledge that we have the right morphology and we have all of these positive stains, it's really looking like we have choroid plexus epithelial cells. And at this point, we can definitely say that the cells that we got from the choroid plexus and are positive for all of these markers, negative for our isotype, or I'm sorry, the true staining for our isotype controls, and it was negative for astrocytes, microglia, and macrophages, that we do know that we've got choroid plexus epithelial cells. So this is our 2D culture. Now, there are some differences between 2D and 3D cell culture, and a lot of our other presenters, if you want to go back and look at some of their presentations, they really did talk about the differences between 2D and 3D culture. So I did want to give you a little bit more of a background on the differences between 2D and 3D. So 2D is simple, easy to do, and cost-effective. However, it doesn't really mimic in vivo, and the cells don't really interact, like they only interact with like the plastic and the edges of the plate. They don't really interact as much with each other in the matrix. They also don't have gradients or a microenvironment. Now, 3D is a little bit harder to do, complex, a little less cost-effective, but it's a better in vivo representation. It can show cell-to-cell interactions and extracellular matrix interactions, and you can also look at gradients and the microenvironment. So this is a really popular model to use in cancer biology and tumor biology, because you can create these little like organoids and actual tumors in culture. So the choroid plexus has a lot of different cell types going on, and we were really just more interested in the barrier properties in that single layer of epithelial cells. We kind of wanted to do a modified 3D. I'm calling it 2.5, because we wanted something simple and cost-effective, but we wanted to be able to represent, the in vivo representation of the barrier properties of the movement and the gradients across this choroid plexus epithelial cell barrier. So we decided to work on this modified 3D, and we used transwell inserts in these petri dishes where we actually plated cells in these transwell inserts in the center, and we could add media on the outside wells and the inside wells. So I kind of, at this point, wanted to see diffusion properties and whether media could move across, if we could form a barrier and if the media could move across said barrier. And as you can see, I did a little bit of a lower level of media on the outside well, and in the inner well, you can see how the media is a little bit higher because I added more into that center well. As you can see here a little bit, I didn't take a really good picture. You can see how the media levels have actually leveled out. So there was movement of media across, and they leveled out. So both the media in the inner and outer wells were at equal levels, indicating that there was permeability and diffusion of this liquid across. But we really wanted to know how good these barrier properties were. And you can't really do that by just looking at a microscope at these transwell inserts. You can't really look at the properties of this barrier. So that led us to working with our next thing, and this is the xCelligence model. I did just want to clarify at the very beginning that Thermo Fisher and Gibco do not endorse xCelligence or its owner, ACE Biosciences. This stuff is presented because it's how I use the model, how data was analyzed, and it's a very specific device, and you'll see as I'm going forward on why I just had to present this data in this system. But I did want to clarify that in the very beginning. So xCelligence is a real-time label-free cell monitoring system. So you've got these little 16-well plates, and they've got electrodes in the bottom of these plates. The picture on the left is a schematic of one of the wells. Now you can run an electron flow through the well, and it'll move from the negative to the positive terminal, and when there are no cells present, there's no impedance. So you have a nice unimpeded electron flow. When you add cells, moving to the right picture, and the cells attach and adhere to the substrate, you start to get an impeded electron flow as the cells attach, making it harder for the electron current to move from the left, the negative terminal, to the positive terminal on the right. So that's the model design with it. And one of the readouts that you'll see from the xCelligence device, which is taking this data and using the initial cell index with which you plated, it will calculate, using the impedance too, it can calculate the cell index and the increases you'll see based on the cells you've added. So this is an example of what a trace would look like on the device. So when you first add the cells, you'll see this rapid increase in the trace as the cells begin to adhere and impede the electron flow. You'll start to have a slower increase due to the cell proliferation kind of in the middle, especially if it's a slower growing cell line. And then you'll get a plateau once the cells have reached confluence and it really blocks the electron flow through the culture. And that is your baseline cell barrier. You can then add different experimental treatments. In this case, they added an apoptosis inducer. And as you can see here, when you see that stark drop in the trace, it's a decrease due to cell death or detachment, and that can actually show you barrier properties. So at the top, when you have the plateau, you've got a nice tight barrier. When you start to see the traces drop off, it's indicating that there is damage to the barrier, whether it's due to cell death or detachment. Something is disturbing the barrier properties. So here is an example of one of my models. This is just an example of an experiment. This, again, it's real time. It's a super nice feature that I could put the cells in, let it grow over time, go home and sleep, come back, work on it the next day. So you can see at the bottom on the y-axis, this is done in hours, and this is about 160 hours. So we went out several days with this specific experiment. And as you can see, there's an example of the plateau when our experiment reached confluence. And then here, these little spikes are when I pause the machine to pull out the plate to add whatever experimental conditions. So those are what those little peaks are moving forward. So here is a zoomed in picture of our barrier and our confluence. As you can see, you see that rapid cell growth as you go from the zero when there are no cells attached and adhered to rapid growth when they are adhering, and then you have that plateau. This is a really great, nice plateau showing that the cells had attached and adhered bottom of the plate. And you have some nice barrier properties here, and we actually had created a usable barrier. So our next step, we're kind of curious. So when HIV, going back to the data I talked about before, with HIV, when it first enters the central nervous system, there's some argument about Trojan herd or Trojan horse, but either way, the infected cells cause a weakening of the choroid plexus barrier, which can allow more movement of infected cells into the choroid plexus and into the central nervous system. TNF-alpha is a pro-inflammatory cytokine that can be secreted by both macrophages and even endothelial cells. If an HIV-infected monocyte crosses the fenestrated endothelium inside the choroid plexus in normal immune surveillance and triggers the release of TNF-alpha, or even encounters a macrophage inside the choroid plexus, which triggers the release of TNF-alpha, we wanted to see if that danger signal could then weaken the choroid plexus epithelial cell barrier. This could be one explanation for how infected cells enter the CNS and trigger that increase in movement in cells across the choroid plexus barrier. When you have more infected cells and more damage in the brain, especially the leakier barrier, you're more likely to have a more severe presentation of HIV-associated neurocognitive disorders. That's why this was really important to us to look at the pro-inflammatory challenge. So there's a lot going on in this picture, but the blue and the pink lines at the very top, those are the choroid plexus plus just media control measurements. Now, as you can see, it stays around 0. I baseline this out since that is our control. And the red and green were cell cultures where at the very beginning on the left of the graph, I added TNF-alpha, the pro-inflammatory cytokine. As you can see, both the red and green traces start to drop off. If you think about our graph from a few seconds earlier with the concept of xCelligence, when you start to see that decrease in trace, it indicates that there is cell detachment or death, but a damage to the barrier function. So TNF-alpha did indeed damage our choroid plexus barrier properties. And this little spike here around, it looks like 90 hours, 90.4, it's a very tiny screen. And you see a little, blip where it lifts up and drops off again. The more times you challenge with TNF-alpha, the worse the barrier properties become. So the more pro-inflammatory challenges you have, the barrier really gets damaged. So we did indicate that a pro-inflammatory cytokine could affect our choroid plexus epithelial cell barrier. So the next thing we wanted to do, we wanted to repeat the experiment with a pro-inflammatory challenge, but then we wanted to follow it with an anti-inflammatory challenge. We decided to use steroids since steroids are anti-inflammatory, and we wanted to see if we could reverse the damage caused by the inflammatory cytokine TNF-alpha. So here again, this is a really busy graph, but the pink and blue traces in the very beginning on the left side, so you have the line dividing the graph. If you look to the left side of that, the left side is the TNF-alpha challenge. So in the top part on the left, you see the pink and the blue, and those are the choroid plexus plus choroid plexus plus media, it's just choroid plexus at that point in this graph. And right below it in the red and the green, you see the choroid plexus plus TNF-alpha. Now, once you move past that divider line in the middle and you move to the right, you actually have 4 different conditions, because we took half of the control wells and half of the TNF-alpha wells, and we added dexamethasone, a steroid. Now, as you can see, the top picture is pink, which is choroid plexus and dexamethasone. Blue is just the choroid plexus plus media. Green is choroid plexus plus the TNF-alpha and dexamethasone, and then red is the choroid plexus plus TNF-alpha. And as you can see here, again, red is the worst. That's choroid plexus plus the pro-inflammatory cytokine with no steroidal treatment. But when you look at green, after that middle divider line, once we started adding dexamethasone to those wells, you actually see an increase with each subsequent administration of dexamethasone. You actually see an increase in the barrier properties on the cell index of these wells, indicating that dexamethasone was helping to repair the barrier properties of these wells. Again, blue, that's going to be kind of our baseline. And another interesting thing, when you just look at the baseline, the choroid plexus plus just dexamethasone, just to rule out any other effects of dexamethasone on the cells by themselves, you do see an increase in barrier function. So dexamethasone did slightly help tighten the barrier of just plain choroid plexus cells without TNF-alpha, but it's still really interesting to note that dexamethasone was able to repair some of the barrier function in TNF damaged cells and affected cells. It's possibly this is one explanation why there is, like with the TNF-alpha, why there is some leakiness of the blood-brain barrier or the blood-CSF barrier, and why infected cells could actually trigger the weakening of the barrier. And this is promising because maybe this steroidal treatment or even any other kind of anti-inflammatory treatment might be helpful in patients suffering from HIV-associated neurocognitive disorders. So for some conclusions, HIV is still a pertinent global health crisis. This is due to the long-lived viral reservoirs in which the brain and the choroid plexus are one. The choroid plexus is really important because it's an immune interface with the monocyte trafficking and barrier dysfunction seen in HIV-associated neurocognitive disorders, as well as some other diseases too. So it's really important to study the choroid plexus epithelial cell barrier. And I forgot to mention this way at the beginning, but the reason why my culture is important is because we did this in rhesus macaques. Those are primates, and they're one of the ideal models of studying HIV in animals. And so while they do have some rat and pig choroid plexus epithelial cell cultures out in the market, rhesus macaques are the better model for this specific study with HIV. So that's why we were set about to create this choroid plexus epithelial cell barrier. Again, choroid plexus epithelial cells are the last line of defense, especially in HIV infections. So we were curious in creating an epithelial cell barrier. And so we created a 2D choroid plexus epithelial cell culture. We validated that by the pathology, the choroid plexus epithelial cell markers, and tight junctions. Also, the choroid plexus epithelial cell modified 3D culture allows us to study the barrier functions of the choroid plexus in real time. We were able to see that the pro-inflammatory TNF-alpha impairs choroid plexus epithelial cell barrier, potentially explaining why we have increases in infected cells into the central nervous system. Also, the anti-inflammatory dexamethasone returns barrier function after pro-inflammatory challenge. So some future directions, I want to continue to work on this modified 3D cell culture model, especially in terms of HIV, and actually look at HIV's effects on this model and see if there are other applications to other diseases, other conditions, other viruses, bacteria, etc. I also want to study the permeability more and see maybe drug diffusion, things like that. This work is in revision in Frontiers, so I'm super excited about that. And then hopefully from your learning objectives, you were able to understand the history, development, and function of the blood-CSF barrier, the choroid plexus, and understand the pathology and therapeutic potential of the choroid plexus. It's a really understudied region of the brain, and it's obviously my favorite part of the brain. So I do hope you can appreciate it a little bit more now. And then also too, how to design a 2D and modified 3D choroid plexus epithelial cell model system. And I'd like to acknowledge everyone at the Primate Research Center who really helped out with this project and focal core, things like that. Dr. McLean's lab, who was my PI for my PhD, as well as Marcella Kuroda and Moon-Ki Kim, who were advisors on my PhD. I'd also like to thank Thermo Fisher and Gibco Scientific for selecting me for this honor, and thank you so much for Chelsea and Chrissy for all of their hard work in getting this together and making sure I met my deadlines and turning things in. I'd also like to thank my current PI at LSU, and then all teachers and professors I've had in my training who've really gotten me to this point in my career. So thank you. Again, thank you guys for your time, and if you guys have any questions, I will be happy to take them.
Thank you, Dr. Delery, for your informative presentation. We will now start the live Q&A portion of the webinar. And if you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. Now, those questions we are unable to answer live today, and those submitted during the on-demand period will be answered via the email address you provided at the time of registration. Okay, Dr. Delery, let's get started. Our first question. What kind of neural progenitors does the CP produce?
That is an excellent question. So there's been a lot of papers out recently talking about neural progenitor niche found in the choroid plexus. And that's still kind of a newer field. Like, they're not really sure what types. They're leaning more towards like radial glial cells, like glial and astrocyte cells. Like, one paper from the group in Japan talked about how they were able to take these neural progenitor stem cells from the choroid plexus and implant it into a damaged spinal cord, and these cells actually differentiated into astrocytes. So they were thinking it turns into either astrocytes or glial cells, depending on the signals from the surrounding area, and like danger-associated molecular patterns, things like that, can actually trigger the differentiation into different cell types depending on the need from the central nervous system. So that is that question. There are a lot of really good papers out there too, if you're interested in reading more about it. They also argue there's age-dependent markers that can affect this, and that the neural progenitor stem cells decrease with age. And a really good paper is Age-Dependent Niche Signals from the Choroid Plexus Regulate Adult Neural Stem Cells by Vargas et al., if you'd really want to read more about that. So next question.
Yes, thank you, Dr. Delery. Is it barrier function or cell population that you are looking at?
So originally we were looking at cell population. We wanted to look at the epithelial cells, and with that led to more barrier function to see, since that's like the last line of defense between pathogens in the central nervous system, they first have to cross the fenestrated capillaries. And since we're discussing this, I do want to apologize quickly. There were some hiccups in the slides, especially when it's showing the choroid plexus, just so you guys weren't able to see what it looks like. So with this question, when we're talking about barrier function, let me pull up that so you can see it too. There's just a glitch in loading the animations. So as you can see here, it's lining the ventricles of the brain. And then also here's an example of the fenestrated capillaries and choroid plexus. And again, on the next page, which looks more at the barrier function. If you look right here, next. Okay, so here you see the fenestrated capillaries and then choroidal epithelium. Now, for a pathogen to get into the central nervous system, it's got to first cross the fenestrated capillaries in the center, go through the stroma and pass through all the immune cells of the stroma, and then exit the choroidal epithelial cells into the CSF in the ventricles. And so that's why we started looking at barrier function more, because it's kind of like the last line of defense before moving into the actual central nervous system. But we are kind of hoping to use this more as like a true 3D model and make more of a stromal-like layer on top, but we're still playing around with that. So hopefully we'll be able to do that in the near future. Next question.
Yes, now this question's two parts. How did you control for contamination? And if your plates got contaminated from necropsy, what did you do?
Great question. Okay, so necropsy is actually that's basically an animal version of an autopsy. That's when you're dissecting the animal. And it's a very, I'd say, dirty environment. While we do wear PPE and we are protected, we have a dedicated suite for the necropsy of the primates. You have multiple pathologists and vets in there pulling out different tissues for different testing types, and it's possible to get fecal matter from one area of the body up into the choroid plexus when you're trying to pull it out of the brain. And so it's very easy to become contaminated with bacteria. So we controlled for contamination by, one, having as sterile an environment as possible, wearing the proper PPE, and extracting the choroid plexus first before doing other parts of the necropsy for collection of tissue. We processed it in a sterile hood and made sure we sterilized it every time we used it. And then we also, if it seemed like it was getting contaminated, we added additional antibacterials or antimycotics like primosin and fungizone if we really needed to supplement the media to make sure contaminants didn't grow. So that's a great question.
Now, Dr. Delery, how is the choroid plexus taken?
Perfect. So like I mentioned with necropsy, they cut the skull open and you're able to pull out the choroid plexus. It looks like this kind of like red string inside the brain, and back in the back by the occipital lobe, you're actually able to see part of it. And a pathologist, we have veterinary pathologists who are skilled at the brain and different parts of the body, and they were able to pull out the red string of the choroid plexus and put it into a tube of PBS with us. Next question?
Yes. Now, why not study TEER in order to study barrier function in terms of resistance the endothelial layer poses to systemic circulation?
Okay, so that's an excellent question. We looked at the epithelial layer rather than the endothelial layer, and basically I was a graduate student, so I used what we had available to us, and at the time the primary center did not have TEER set up, so I used what was available to us and what we had supplies for. But it's a great question on a technology I could use in the future should I have access to it. But I was basically using previously studied models that my PI had used and equipment he had already, because I didn't really have time to write a grant to get more funding from the NIH for more equipment. But that is an excellent suggestion for future testing.
Dr. Delery, can you elaborate on why you couldn't use human choroid plexus?
That's awesome. Okay, so you can't use human choroid plexus because the only samples I get, I don't really know anyone out there who would voluntarily submit to an open brain surgery where we would remove pieces of their choroid plexus. And they do actually have, I believe, a human line out there. However, it was taken from a patient who had cancer in his choroid plexus, and that can affect growth rates of the cells and behavior of cells. So we didn't want to use that just because it might have different properties than a true normal choroid plexus since it was cancerous. So that's why we stayed with the next best model, the primate model.
Thank you, Dr. Delery. Now, after extracting the choroid plexus, do you dissociate it into individual cells before culturing, or do you put the whole string in the media?
Excellent question. So yes, actually we did. We mechanically dissociated it. We chopped it up with a scalpel until we had little pieces of it. And then we also used a digestion collagenase disc-based solution to help break it up a little bit more. It was easier if cells attached in clusters, because we didn't dissociate it really well, they actually grew a little bit better. But if you put whole choroid plexus chunks in there, it also just didn't grow well. So it's kind of that fine line in between. And we do have a paper coming out that's currently in revision that will have a more detailed methodology too. So if you're interested in that and you want to follow up with me, it should hopefully be out in the next couple of months, or shoot me an email. It was on the very first slide. Let me pull it back up right now, actually, so you guys can get it. You're more than welcome to submit your questions through Thermo Fisher and Gibco, or email me and ask me any questions, or if you just want to say, hey, send me the paper when it gets published, and I would be more than happy to do that.
Thank you, Dr. Delery. Now, we've got some great questions coming in. We have time for a few more. And let's go with this one. It's come in several times. How many replicates were done with the xCelligence studies?
Oh, awesome question. So for xCelligence, we're lucky in that it's a 16-well plate. And as you remember from some of the pictures, let me pull up so I can show you right now. You can see 4 lines here, and obviously 16 divided by 4 is 4. So we had 4 separate wells for each condition, and they were averaged together to produce these lines. Kind of when you see this, you see the long vertical lines here, that's actually the standard deviation on the graph. They were a little bit messier for some of the other ones, which is why I wanted to do just the lines themselves. But that's 4 replicates for that one right there. And I know replicates are kind of an iffy subject, because do you mean replicates of animals, or do you mean replicates of studies? We repeated the TNF-alpha studies and dexamethasone studies with a couple other animals too. I believe there's 3 with TNF-alpha and 2 with dexamethasone. So in addition to multiple wells for each animal, we did have more than 1 animal to practice it in. Again, a lot of people who work with mice are going to be upset by the fact that we are only able to do 5 primates for all of these studies. We've done more since then, but just for this the paper topic we did about 5 primates, just because they are a select resource and more scarce than mice, and also way more expensive to house for a year than mice. So we used our available resources and an appropriate N for primate studies.
Thank you. Now, our next question, how are you using this in your current studies?
Oh, that's awesome. Okay, so I've kind of moved a little bit from the HIV studies. I'm now studying neurobiology of alcoholism and inflammasomes and opiates in the brain. And actually, as you know, alcohol and opiates have to cross the central nervous system or cross the blood-brain barrier and Blood-CSF barrier to enter the central nervous system. And so I'm working that into my PI's grants and also submitting one of my own grants later this year to start looking at the choroid plexus more in relation to other conditions and other disorders. And having a primary cell culture that allows me to do additional testing on it is fantastic. And we have some frozen cultures too that we're going to work with in the future. So I'm excited to see if I can expand on these barrier properties to study opiates, alcohol, things like that.
Thank you, Dr. Delery. We're excited to see this too, and we'd love to know what do you hope to accomplish with this?
Ideally, so I know this is a really tough topic for a lot of people, but animal research. You know, if you're familiar with the U.S. IACUC laws, the goal is to reduce, replace, and limit the number of animals you use, replace it with more simple models, because we want to make sure they have the most humane care and we're not causing undue suffering or anything like that. And while I realize it's a necessary step in pharmacological testing, you know, disease modeling, things like that, I would like to help create ways that we limit the number of animals that we use. And so I hope that this culture can be used to study conditions ex vivo that would also limit the number of primates that would have to be experimented on, whether it's, you know, drug delivery or the effects of drugs on the Blood-CSF barrier, et cetera. I'd love to use this model to help you know, humankind increase quality of life, but also limit the amount of animals we use in studies. So that's my hope for this model going forward.
Thank you, Dr. Delery. Now, do you have any final comments for our audience?
No, thank you guys so much for tuning in today to watch this. And again, you know, please reach out to me on, you know, my email. Again, it's listed right here too, and then social media. I would love to know things you want to learn about, things you want to talk about. If you're watching and you're applying to grad school soon, feel free to let me know if you need any help or advice going forward or picking a dissertation topic or a PI or whatever. I'm still fairly new to the postdoc game, so not too far away from the PhD that I can't provide advice if you guys need it. And again, just get out there and love science, love what you guys do, and feel free to reach out to me and connect with me on Instagram or LinkedIn. I believe I'm the only, or the top Elizabeth Delery on LinkedIn right now, so feel free to reach out to me on social media.
A big thank you again to Dr. Elizabeth Delery for her time today and her important research. I'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Now, before we go, I want to thank our audience for joining us today and for their interesting questions. Again, just a reminder, those questions we did not have time to answer today and those submitted during the on-demand period will be addressed by Dr. Delery via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you shortly via email when it's available for replay. We encourage you to share that email with all your colleagues who may have missed today's live event. Thanks for joining us today, and we hope to see you again soon. Bye-bye.